Greetings, everybody. My name is Arianna, and I'm going to introduce you with some concept about spatial proteomics, in particular imaging mass cytometry and its application in the study of the alloimmune microenvironment in liver transplant rejection. The aims for this seminar is to identify the advantage of imaging mass cytometry in the study of liver allograft rejection, and how using image preprocessing actually enables and enhances data analysis, and finally discuss the identification of biomarker of rejection that can actually help the clinician in making diagnosis of rejection. Liver transplantation is the standard of care for patients with end-stage liver disease, and today we reached excellent outcomes for graft and patient survival. However, acute T-cell mediated rejection remains the most frequent complication in both pediatric and adult liver transplant recipients, and you can see here that children can experience acute rejection up to 60% of cases at 5 years post-transplant, and in the adults population, this can reach 50%. Today, the diagnosis of rejection relies on liver function test abnormalities, which prompt to a liver biopsy, and the tissue obtained from a core needle biopsy is stained with hematoxylin and neoxin, and a pathologist can visually assess the presence of rejection and then assess the severity of rejection by using Banff criteria. When we analyzed our own institutional data that included more than 200 pediatric liver transplant recipients and more than 800 biopsy performed either for surveillance or for cause, we saw that the level of liver function tests were not really associated with the severity of rejection. And more importantly, when we look at the ability of these liver function tests to predict rejection, their accuracy was pretty low. Finally, 18% of surveillance biopsies performed in children without signs symptoms of rejection harbored histological evidence of mild or moderate rejection. Previous studies have used mainly bulk RNA sequencing or FFPE immunofluorescence technology to try to better characterize the immunomicroenvironment within liver allograft rejection. As you can see here, there are some markers, but we don't have a high level of details associated with them. And even when they use whole genome macroarrays to try to identify a set of genes that can help to differentiate between rejection and no rejection, they couldn't find any. So this is why we need some single cell technology that can help us to better characterize the allograft immunomicroenvironment. Recently, there is this study where they use multiplex immunohistochemistry to try to analyze the cell-to-cell interaction. They have characterized the segregation of markers on the cell surface, but again, the analyses were limited to interaction between just two cell types. What are the advantage in using imaging mass cytometry? IMC is a multiplex imaging technology that can use more than 40 markers simultaneously, and this allows a deep phenotyping and characterization of immunocells. Additionally, we can characterize the activation states by using RNA probes. We can also have information on the signaling pathway, And we also can study the spatial relationship among different cell types. Usually, we have FFP slides that are stained with a cocktail of antibodies conjugated with metal ions. Those stained slides are then ablated using UV laser, and the ion clouds is then analyzed by mass spectrometry. However, there are some cons associated with imaging mass cytometry and in general all multiplex imaging technologies that are related to the image artifacts. In particular, those are due to channel spillover, noise, antibodies aggregation, shot noise, and in general, they are due to antibody concentration and arrangements. So what we can do to reduce these image artifacts? First of all, it is important to have an antibody panel that is well designed. And within this panel, antibody needs to be accurately titrated. And finally, we can use pre-processing tool on raw image to enhance the quality of our image. How do we recognize artifacts? This comes from our own data. And as you can see here, there are some strange association of markers. We have some FOXP3 that is associated with CD68 and some cluster that co-express CD66, CD20, and CD28, that it's usually a T-cell marker. So in theory, we have two choices. Either we discard those cells or we can force those cells in one phenotype. So we assign a phenotype to those cells with the risk of whoever assigned the wrong phenotype for those unclear clusters. With our data, we went back to the raw signal and we realized that there was a contamination, a spillover of CD68 into FOXP3. And we used this pipeline called MAUI that was developed by Baranski et al. And it's a MATLAB-based pipeline where using 3 steps called background removal, denoising, and aggregates removal, we can actually improve the signal of the of a specific markers. In particular, you can see here that after background removals, meaning subtracting CD68 from a non-CD68 channel, we are getting rid of the improper signal. Following, there is this denoising step where we are eliminating this unspecific signal. And lastly, there is the aggregates removal where these small dots that come from aggregates of antibodies are eliminated. And we can actually repeat these 3 steps for all the channels, thus for all the markers that we have in the panel. This is an example of how this preprocessing is actually enhancing our quality of data. You can see here that the CD4 signal is actually improving, as well as CD68 and CD8 compared to the raw signal, non-preprocessed image. And this is translating also in a mask, so in a actual recognition of cell phenotypes that is missing in the initial non-preprocessed data. And this is true for CD4 as well as for CD68. And this actually is a proof that preprocessing is able to facilitate and also enhance the assignment of phenotype within the image. The other difficulty usually associated with IMC is the segmentation of raw images. And there are currently different platforms. The first one they came out was Elastic Plus Cell Profiler. We have also Mesmer and DeepCell, CellSeg, CellPose, among the others. And in our lab, we started with the association of Elastic and Cell Profiler for raw image segmentation. As you can see here, there is some operator variability, meaning that based on how we draw the membrane on the image, it is affecting the appearance of the mask. So we realized soon that this operator variability could actually impact our data. We compared the number of cells obtained by these two operators, and we saw that there were some slight differences. However, we did not go further to estimate whether these different mode in cell segmentation, for example, could impact cell-to-cell interaction. However, we decided to use another approach. We discovered that Boden-Miller Lab put together a Steinbock toolkit based on a Docker container that can allow desegmentation by using 3 different platforms, Elastic Plus Cell Profiler, Deep Cell Mesmer, and CellPose. We choose to use Deep Cell because it is an automated, a deep learning algorithm that allows an automated identification of cell features. And so this is reducing the time that an operator can actually spend in training each image. And the output of the segmentation is always a cell mask for each image or each region of interest, and single cell expression matrix that is then used for downstream analysis. So putting together our steps, this is our complete pipeline, going from acquisition of raw image, pre-processing, segmentation, and finally our data analysis that is performed using RStudio. Now I want to give you some example of how this entire pipeline works, and showing some results of our spatially resolved alloreactive microenvironment in acute and chronic liver transplant rejection. So we selected different sample types, in particular 24 non-rejection cases, 41 cases showing acute T-cell mediated rejection, which included a total of 58 regions of interest, and 14 cases of chronic rejection. Those FFP slides were stained with 22 antibody panels that included most of the immunomarkers and 3 structural markers, such as collagen type 1, CK7 that was used for bile duct identification, and CD31 for endothelial cell. After segmentation of raw image, we obtained a single cell dataset which included more than 400,000 cells. Those cells were assigned to 10 different phenotypes, which included 3 non-immunometaclusters and 7 immunometaclusters, which had a different distribution across the 3 clinical groups: no rejection, acute rejection, and chronic rejection. And as you can see here, hepatocytes was the most abundant phenotype associated with no rejection. We also looked at the distribution of immunophenotypes, or immunoclusters. CD4, CD8, monocytes, B cell, and plasma cell were different across the 3 groups, while there was no difference in macrophage and neutrophils among the groups. We also tried to further characterize each metacluster, and so we performed a subclustering within the CD4 compartment, which led us to identify 9 different subpopulations that, as you can see here, had a different distribution and density across the 3 groups. When we look at the distribution within each clinical group, we saw that resident memory CD4 T-cell was the most abundant phenotype identified in no rejection, while CD3 positive, CD4 positive T-cell were predominant in both rejection groups. Additionally, we saw that there were differences in two activated phenotypes, naive CD4 T-cell and activated CD4 T-cell, and finally, difference in distribution in two regulatory phenotypes, a PD-1 positive CD4 T-cell subpopulation and a Treg subpopulation that had a high expression of HLA-DR. By subclustering the CD8 compartment, we identified 5 subpopulations. And again, here you can see the distribution of those 5 subpopulations within the tissue. And the most abundant phenotype identified was the CD3-CD8 T-cell, while proliferating CD8 T-cell was predominant in TCMR. And finally, we saw some differences in the distribution of 2 PD-1 CD8 T-cell population. In the macrophage compartment, we were able to distinguish 4 different M1 macrophages and 5 M2 macrophages subpopulation. By looking at their distribution across the 3 clinical groups, we saw that proliferating M1 and CD16 M1 macrophages had a different distribution across the groups, as well as HLA-DR M2 macrophages and CD16 positive M2 macrophages. We also studied the cell-to-cell interaction, and here it's pretty evident, as in TCMR, there are a lot of interaction that can be either avoidance, blue line, or attraction, red lines, when compared to non-rejection and chronic rejection. In particular, in TCMR, there is a lot more interaction between effector T-cell and PD-1 T-cell that probably try to dampen the immuno-inflammatory response. Based on subclustering interaction, we were able to recluster those to identify at the end 9 cellular neighborhoods that presented a different spatial distribution across the 3 clinical groups. And in particular, when we compare those cell neighborhoods in terms of percentage, we saw that CD8 enriched and B-cell and monocytes enriched cellular neighborhoods were predominant in TCMR, while CD16 positive T helper enriched cellular neighborhood was predominant in no rejection. We used the subpopulation information to build a predictive model of rejection by using a lasso regression algorithm, which identified the most predictive features of acute rejection. And I want to point here that in both cases, we can see the presence of PD-1 T-cell population as predictive of TCMR. And both models had a pretty high accuracy in predicting acute rejection. So in conclusion, IMC allows an in-depth phenotype of immunocells and characterization of their functional status. An accurate preprocessing of raw image facilitate and enhance the quality of the image and increase the accuracy of phenotype assignment. Use of IMC in combination with image preprocessing enable detection of rare cell population that can be used as a biomarker, in our case, as a biomarker of liver rejection. In clinical liver transplantation, we identify these PD-1 T-cell populations that needs to be further investigated to understand their role in rejection pathophysiology. And the special feature of IMC can provide not only information on cell-to-cell interaction, but also detail on cell-to-cell aggregation that can be associated with a particular state of disease. I would like to thank you, the members of Emamaullee Transplant Lab, in particular, Brittany, Sarah, our biostatistician, and our research coordinator, Carly. Thank you.